Schistosoma mansoni is the causative agent of intestinal schistosomiasis, a neglected tropical disease endemic to areas of sub-Saharan Africa. The current method for monitoring infections in the snail vector depends on microscopy by visualizing parasite shedding. However, this method is prone to missing infections due to the variable parasite development time. Our goal is to develop a diagnostic PCR assay for accurately detecting Schistosoma mansoni DNA in the African snail vector Biomphalaria sudanica. To do this, we addressed 4 questions. The first question, what sample dilutions provide the best PCR amplification? To test different multiplex PCR markers, we used the extracted genomic DNA of Kenyan B. sudanica snails exposed to S. mansoni, half of which were confirmed as infected by schistosomes using microscopy. DNA was then tested at full concentration and at different dilutions with water to dilute suspected PCR inhibitors in our samples. We tested 3 snail primers in combination with the ND5 marker, previously shown to be a S. mansoni specific marker. We found that a 1 to 100 dilution of snail DNA improved amplification for all snail markers. However, 2 of the 3 snail markers showed amplicon variability and problems with amplicon visualization during gel electrophoresis, ruling them out for further development. The snail schistosome marker taken forward still had amplification bias for the schistosome-specific marker during multiplex reactions. To lead on from this, our second question was, can an adjustment of primer ratio improve co-amplification of our PCR primers? We found that having schistosome-specific primers at a 1 to 3rd concentration relative to snail primers worked well for getting equal amplification across markers. Our third question was to determine the limit of detection for this assay. We spiked DNA extracted from laboratory B. sudanica snail that had not been exposed to schistosomes with a dilution series of DNA from a single adult S. mansoni worm. The limit of detection for S. mansoni DNA in the multiplex PCR assay was higher than would be required to detect a single parasite. Therefore, further refinement of the assay is required to improve this limit of detection. Leading to our final question, can an inhibitor-resistant polymerase improve amplification with reduced sample dilutions? We tested fire polymerase on laboratory B. sudanica snail DNA spiked with miracidia DNA. We found that this polymerase reduced the dilution necessary to amplify ND4 multiplex with ND5 from 1 to 100 to 1 to 10 dilution. We then applied this polymerase to screen Kenyan snails used for a genome-wide association study to compare allele frequencies of infected and uninfected snails. 11% of the samples did not amplify our control marker at the 1 to 10 dilution, but was resolved at 1 to 100 dilution. Although the assay needs further optimization, we successfully identified pre-patent infection in negatively infected snails. Moving forward, we are exploring alternative conditions to improve amplification of our control markers without the need for further sample dilution. Thank you.